semester goes by super fast, and there's not enough time to go into all the detail I'd want about lipid and amino acid metabolism, but here are some of the key things to know. First, I have a few things to say about CoA. So, CoA is going to show up a lot of different places in our metabolism. CoA itself is this bigger thing that's derived from vitamin B5 or pantothenate. It has this big kind of handle that you can use to have specific binding sites that will recognize this CoA and hold on to it. But really, the important part is just the sulfur right here. Why is it important? Well, sulfurs are swappable. Sulfur is right underneath oxygen on our periodic table, so same column but different rows. Being below oxygen means that sulfur basically has an extra shell. Its electrons are going to be further out. So although it has the same number of valence electrons, those valence electrons are going to be more reactive. So sulfur is going to be more nucleophilic. It's also going to be, because it's bigger, it's going to be harder for sulfur to form effective covalent bonds. Remember that covalent bond formation involves the kind of overlap of electron clouds and orbitals. And well, when you have a sulfur trying to bond with something smaller, then you're kind of in a situation where you have someone with a really, really big hand trying to shake hands with a little kid. It's going to be hard. So those thio bonds to this sulfur are going to be weaker. They're going to be easier to break. And sulfur is going to often be the one breaking those bonds. So you can have sulfurs be swapping each other out, as well as have sulfur kind of make for a vulnerable point at which you can do things like form esters, form amides, and form carbon-carbon bonds. In the case that we saw before, we saw how pyruvate can get converted into acetyl-CoA, where we had an acetyl group put on CoA. Acetyl group is just one form of an acyl group that we can stick on CoA. So we'll see later how we'll have fatty acyl-CoA, where basically we stick a big fatty acid onto a CoA group. An acyl group, remember, is just where we have a carbonyl carbon attached to some hydrocarbon group. And then in the case of our CoAs, our acyl-CoAs, this is then going to be a CoA over here. So we saw how we could basically take pyruvate and we could transfer it to CoA and enter the tricarboxylic acid cycle that way. Let's also, though, today talk about what else we can do with acetyl-CoA. One of the other things that we can do with acetyl-CoA is actually use it to generate lipids. So we'll talk about lipid synthesis. We'll also talk about how lipids can be broken down to give you acetyl-CoA. And we'll talk about how some proteins can also be broken down to give you acetyl-CoA. So acetyl-CoA is really kind of central to a lot of different things in metabolism. And let's dive in. First, let's talk about where things are happening. Remember that when we talked about glycolysis, that was happening in the cytosol. And we talked about the citric acid cycle, that was happening in the mitochondrial matrix. In the case of fatty acid synthesis, the synthesis itself is going to occur in the cytosol, and it's going to happen predominantly in our liver and adipose tissues, so our fat cells. The breakdown of fatty acids happens through a process called beta oxidation, and that's going to be happening in the mitochondria. What happens is that your liver and your fat cells, they're able to synthesize these lipids, and they store them in the form of triacylglycerides. So remember, that's where we had 3 lipids on a glycerol backbone, and that was going to be very hydrophobic, allowed us to kind of like store these fats in these lipid droplets that aren't going to hog up water, be this very compact form of storage. When your cells have excess energy, then your liver and fat cells will build up more of this fat and store it. When other tissues need that energy, basically those fatty acids can be delivered. Instead of delivering just those globs of fat, they're typically delivered by chopping off the fatty acids, so by like lipases, and then transferring those fatty acid tails held onto proteins in the bloodstream, such as serum albumin. These fatty acids can then travel to the tissues can take them up, and they can do things with them. They can break them down, or the tissues can also incorporate them to make things that they need. Maybe they need membrane lipids, so our like phospholipids. incorporate them into more complex lipids, which occurs in the ER, or the endoplasmic reticulum. That's where we're going. Let's start, though, with just talking about this lipid synthesis. What do we need to do to make a lipid? Well, to make a fatty acid, we're going to need acetyl-CoA. We'll talk later about how this isn't as simple as just taking that acetyl-CoA out of the mitochondria, because acetyl-CoA itself can't get in and out of the mitochondria. So we're actually going to take citrate out of the mitochondria, which we can then convert to give us our acetyl-CoA, which we can use to make our fatty acids. But in order to make lipids, well, other lipids, if we want, say, a membrane lipid, we're going to need to attach it onto a glycerol backbone. We also need to attach it onto a glycerol backbone in order to make our triglycerides. So remember that we had the glycerol backbone, it had those 3 OH groups. In the case of our triglycerides, we're hooking up 3 of these fatty acid tails, 1 per OH. In the case of our phospholipids, we'd hook up 2, and then we'd have the phospholipid head. It turns out that even if you want one of those triglycerides, you start with having glycerol 3-phosphate, so you have 1 phosphate group there already. And this glycerol 3-phosphate is coming from dihydroxyacetone phosphate, or DCAP, which remember was coming from glycolysis. So this was basically, this component comes from glycolysis, this component comes from the TCA, and so fatty acids or lipid synthesis is kind of at the intersection of both of these, and we'll see how it can kind of be regulated by both of these pathways as well. Acetyl-CoA can also be used to make cholesterol, other steroids. We're not really going to talk about that, but know that there's this enzyme HMG-CoA, which is really important for this. And this is the target of statin drugs. But let's go back and let's talk about how we make these fatty acids. This fatty acid synthesis, it gets a little complex looking, and so don't worry about the chemical details. The important things to know are one is that this is happening in this complex called the fatty acid synthase. In our bodies, this is actually carried out by one big polyprotein that has all these different enzymatic sites on it. So there's going to be a bunch of chemical reactions happening, but all of these are happening on the same enzyme, the same protein. Whereas in like bacteria, it's going to be broken up into different proteins. What's going to be happening is that we're going to be synthesizing these fatty acids 2 carbons at a time. When we talk about 2 carbons, well, you're probably thinking acetyl-CoA's got 2 carbons, right? So let's just piece a bunch of acetyl-CoA's together. Somewhat confusingly, you're actually taking 1 acetyl-CoA, and instead of adding another acetyl-CoA directly to it, you're adding this thing called milanoil-CoA. So if we look at milanoil-CoA and we look at acetyl-CoA, we can see that milanoil-CoA has got this extra carboxylic acid group. So it has an extra carbon, so it's a 3-carbon compound, but we're only going to add 2 of the carbons. So we're going to use energy in order to add this carbon dioxide, which we're then going to remove. This might seem kind of silly, but what's happening is that form a carbon-carbon bond. It's not going to be energetically favorable, so we're going to have to link it to something that is. What we're doing here is basically we're spending this ATP in order to activate this carboxylic acid that we add on. When we add it on, we add it in the strategic position where we have a beta keto acid. So we have this carboxylic acid group with a ketone that's going to be in the alpha beta position. As we discussed the citric acid cycle, decarboxylation of a beta keto acid is thermodynamically favorable. And so this is going to have the energy, provide the energy that we need in order to join these carbon groups together. The carboxylation is happening with this acetyl-CoA carboxylase, and this is going to give us our milanoil-CoA. So we would have acetyl-CoA and we'd have milanoil-CoA, but we're not actually going to join them as acetyl-CoA and milanoil-CoA. Instead, we're first going to link them onto the protein. First, let's orient ourselves to where things are happening. So remember, this is happening in the fatty acid synthase, and it looks a bit like this. It's got this key part that's going to be this acyl carrier protein that's going to be the point of attachment for our milanoil groups. And then it's going to have over here, it's going to have a cysteine residue. And it's basically going to transfer the milano groups from this ACP to that cysteine residue. And the cysteine residue, well, we're going to have our growing chain. Our growing chain is going to be held here, and it's just going to keep getting longer and longer and longer until finally we get that final chain. But the incoming groups are bound to this acyl carrier protein and then moved over. And so you're building kind of from the bottom up. And the newest added ones are actually going to be closest to the carboxylic acid group when we go and we cut this off. But going back to how it's attached, it's going to be attached through one of those sulfur linkages. So before we were talking about how we had coenzyme A, and it was really great because it had this big sulfur, and sulfurs are swappable. And we actually have our Milano-CoA and we have our acetyl-CoA, and we want to transfer that part, that swappable part, onto a different sulfur. And so in the case of a cysteine, yeah, we just attach things to the sulfur and the cysteine. In the case of the acyl carrier protein, it's actually not that part of the protein itself, but rather what the protein is holding on to that plays a starring role here. Remember CoA, how it basically, all we really cared about was the sulfur group, but then it had this big part sticking off that was derived from our vitamin B5? Well, basically what we have here, in the case of our ACP, it's bound to this pantothenic acid group. And so basically it's almost identical to our CoA, except that instead of being bound to a 5-phospho-AMP, it's bound to a serine. So it's bound to a serine in this ACP protein, covalently attached to this group, which has this sulfur that's going to be swappable. And so basically we can take the group from our Milano-CoA, or from initially our acetyl-CoA, and then esterify it to this SH group. So swap out what was attached to CoA now to this ACP. So it's basically a swap for a swap, and of the pretty much exact same thing, but they're just attached in different ways. So with coenzyme A, it was free floating, whereas here it's attached to the protein. And when it's attached, well, now it can get shifted, it can get pushed over. So you get it initially attached to the ACP, then transferred over to the cysteine residue. Now you just keep doing that over and over as you bring the new groups in, transferring them over. With each transfer over, you're going to lose a CO2. You're going to use the energy from that favorable decarboxylation in order to drive this process. When you're driving this process, well, now you're also going to have to be reducing things along the way, because remember, we want our fatty acid. We're trying to make a saturated fatty acid. It's going to be fully reduced. So as we add this, we're going to then have to go from a carbon dioxide to a fully saturated, so like an alkane. This is going to require 2 reduction steps. And to do this, we're going to use NADPH. So remember that NADPH was similar to NADH, except that it had this extra phosphate group. This phosphate group was allowing us ourselves to kind of distinguish between these 2 molecules. And this was important because, well, we wanted to use that NAD typically for our oxidative reactions, things like glycolysis and the citric acid cycle. In order to have enough of that NAD plus in that form, we would need to keep a high concentration of NAD plus compared to NADH. This was going to make it so that we could have those reactions be thermodynamically favorable. But in the case of our fatty acid synthesis, this is going to be a reductive reaction. So we would need to have NADH levels high. But we wouldn't want NADH levels to be high if we want to have this NAD plus ready to use to keep those other reactions favorable. So remember that glycolysis and this lipid synthesis are both happening in the cytoplasm. And so how are we going to rectify this? We're going to use NADPH instead of NADH. We can keep a high concentration of NADPH compared to NADP+, and therefore we have this pool that we're able to use. Basically, we just go through these reductive steps to get this saturated. And then we just keep doing this over and over and over. Well, not forever. Instead, this synthase is going to stop at this kind of like default point of palmitate. So the 16 carbon fatty acid, at which point palmitoyl thioesterase is going to hydrolyze it off, leaving us with this fatty acid, this palmitate. If we wanted a longer fatty acid, we would have to go through an elongation reaction that could happen in the ER. And also in the ER, we could do things like introduce alkenes, so introduce like double bonds. And in those cases, basically, you would still make the initial fully saturated form and then introduce the carbon bonds. So the key players here are going to be our acetyl-CoA carboxylase, which is going to be producing the milanoil-CoA, and then the fatty acid synthase. In terms of the acetyl-CoA carboxylase, this is going to be an important regulatory step because milanoil-CoA is going to serve as an important regulatory molecule. If we think about milanoil-CoA, when would we have a lot of it? Well, we would have a lot of it if we're making fatty acids. If we're making fatty acids, we don't want to break down the fatty acids that we're making. So as we'll talk about later, the fatty acid synthesis and fatty acid breakdown are going to be inversely regulated, similar to how we inversely regulated glycolysis and gluconeogenesis. In this case, milanoil-CoA is going to inhibit this CPT1 that we'll talk about, which is actually how we get fatty acids into the mitochondria for beta oxidation. But for now, we've generated this fatty acid. But first, a complication, because, well, we needed acetyl-CoA to start that all. But acetyl-CoA can't enter or leave the mitochondria. We talked about this when we discussed how when we did the oxidative decarboxylation of pyruvate, we did it inside of the mitochondrial matrix because that CoA couldn't get through. So instead of trying to transport this acetyl-CoA through, we're going to transfer something we can convert into acetyl-CoA. So remember, we're generating this acetyl-CoA in the mitochondrial matrix, and now we want to get it out. How we could get it out is in the form of this citrate. So remember that in the tricarboxylic acid cycle, we combine acetyl-CoA, we combine the acetyl group with oxaloacetate to give a citrate. We can take this citrate out of the mitochondrial matrix, and then we can use that citrate to basically reverse things to separate it back into oxaloacetate and acetyl-CoA, and then we could use the acetyl-CoA. When we're doing this, we can do it using this enzyme ATP citrate lyase, which will produce our acetyl-CoA and our oxaloacetate. As we discussed when we were talking about the TCA cycle, oxaloacetate can't get in and out either. It's going to be highly regulated. And so instead, we're going to need to, if we want to get that, the rest of the carbons back into the matrix where they can be broken down further, oxidized further, we're going to need to transport them in a different form. We have kind of two options. We can convert it to malate or we can convert it to pyruvate. Either way, we're actually going to convert it into malate first. This is the same reaction we were looking at when we were talking about how we get NADH equivalents into the mitochondrial matrix via the malate aspartate shuttle. Remember there, we took oxaloacetate and we took NADH and we reduced oxaloacetate to malate. Malate was then able to sneak those electrons into the mitochondrial matrix where they could be transferred back onto NAD plus to give us NADH. The reason why we were doing it there was basically to regenerate that NADH inside of the mitochondrial matrix. That's not really our goal here. If you have malate and you send it through, you're going to get more energy than if you send pyruvate through because malate's got more carbons in it. But what if what you care about is not so much those carbons, that energy? You've got plenty of that energy if you're making fats probably. Instead, you're going to want that NADPH. And so what you want to do is basically take this malate and you're going to oxidize it but you're going to oxidize it using NADP plus to give you NADPH. This takes place with this enzyme, malic enzyme. You're getting this NADPH, which you can use to power the lipid synthesis. That's not the only way we can get NADPH. Another key way is going to be the pentose phosphate pathway. The pentose phosphate pathway was one of our options for glucose that entered the cell. And basically it can give you different things, including pentoses that you can use to make nucleic acids, but it could also give us lots of NADPH. This NADPH can then be used for lipid synthesis. And so by doing this pentose phosphate pathway, we can then generate fatty acids and other lipids. Let's now talk about how we can break down fatty acids. So fatty acid breakdown or catabolism happens in the mitochondria, and this is happening through a process called beta oxidation. It's similar to the reverse of what we were looking at before, in the case that we are breaking down things to, acetyl groups that are 2 carbons at a time. But note here, we're not having a malonyl-CoA intermediate. So when we're building up a malonyl-CoA, that's really indicating that we're synthesizing lipids. Don't worry about the details of these reactions, but basically before we had a bunch of reductions, now we have a bunch of oxidations. How this is going to happen is basically we're going to be doing these oxidations using both FAD and NAD+ to give us FADH2 and NADH. Remember that this is going to be happening in the mitochondria, so that NADH is already going to be where it needs to be to enter the TCA, so it can enter and get the full ATP yield at complex 1. The FADH2, well, here it's going to contribute to our Q pool, but it's not going to pump proteins from that first step. So you're going to get less energy from that FADH2 than from the NADH, but that's just a technical detail. And you're still going to get a lot of energy from fatty acid breakdown. How this breakdown goes down is that basically you're going to do this first oxidation to generate this alkene, this carbon-carbon double bonds. Then what's going to happen is you're going to hydrate that. You're going to add water across the bond. You're going to then oxidize that alcohol group to a ketone. And then you're going to cleave it off. And the way that cleave off the acetyl group, the way that you cleave off the acetyl group is going to be by using a CoA. Remember that our sulfur groups are going to be swappable. So basically we have this, this fatty acid. It's actually in the form of this fatty acid CoA, this acyl CoA, which I'll discuss in a minute. And now what you're going to do is you're going to take another CoA. So when you chop it off, it's kind of like you're chopping off part, but then having your scissors stuck to where you cut. So this sulfur linkage basically is going to come and it's going to attack this carbonyl carbon. You're going to get a bond to that carbonyl carbon, and then you're going to get this end group is going to get cut off. When we were building before, we were building from this from the end that was attached to the protein, to the ACP. And here we're going to be removing from that same end, but here we're attached to CoA. And we're going to be cutting off these CoA pieces one at a time. And remember, each of these CoAs is 2 carbons. So what's going to happen is that we're going to be, if we have an even number of fatty acid, we're eventually going to break it all off into acetyl-CoA. If we have a odd number of fatty acids, which are rarer, but they do occur, then what's going to happen is you'll end up with a 3-carbon compound that's called propionyl-CoA. And that can basically be carboxylated to give you a 4-carbon compound that can then get broken down to give you those 2 acetyl-CoAs. So we can handle it. We can also handle if we have saturated fatty acids or unsaturated fatty acids, that is. And so remember that our natural unsaturated fatty acids are going to be cis double bonds. And in the form, and what we're dealing with here is actually going to be a trans double bond that we add in order to break things. So those cis bonds will get converted into trans bonds, and then they can be cut down that way. But you're going to get a little less energy because you don't have that initial oxidation in order to make the bonds. But this is how we're going to be breaking down the fatty acids, and that's going to be occurring in the mitochondrial matrix. But there's a problem. because that's fatty acids can't get carried in as in the form of the acyl-CoAs. So in our cytosol, basically what's going to happen is that we can have these fatty acids attached to to a CoA group. And so when you do this, it's done by this like fatty acyl-CoA synthetase. And it's going to take the fatty acid and it's going to put it onto it's going to put it onto CoA. It's going to take ATP to do this because we're forming, remember, this higher energy bond. And so now we have this fatty acyl-CoA, but it can't, this was in the cytosol, and it needs to get into the mitochondrial matrix. We can't take it in as is, and so instead we're basically going to use this one higher energy bond and swap it for another higher energy bond, but this case on the carnitine. So carnitine is going to be another one of these small molecules, and it's going to have a group that can attach to this, to where the CoA was attached in a swappable manner. This will give you a fatty acyl carnitine, and then it can get transferred through this translocase into the mitochondrial matrix. Here, you're going to basically have another carnitine acyltransferase, which was the thing that is going to transfer it onto carnitine. Now you're going to transfer it off of carnitine and onto the CoA. This will give you a fatty acyl-CoA, and that can then be broken down, and then those acetyl-CoA pieces can enter the tricarboxylic acid cycle. So what happens is you have this fatty acid in the cytosol. Fatty acyl-CoA synthetase is going to hook that fatty acid onto a CoA handle. It's going to then swap that CoA handle onto from the CoA onto carnitine. This is done with this enzyme carnitine acyltransferase, or CAT1. Sometimes this enzyme is also called CPT1. So basically the CAT1 would stand for carnitine acyltransferase. But remember, our default fatty acid was our palmitine. So we have palmitoyl transferase, carnitine palmitoyl transferase. So you also see it called CPT1. In terms of the 1 and the 2, well, basically, because you need one version on the outer mitochondrial matrix and one version on the inner mitochondrial matrix. So CAT1 is on this outer, and then CAT2 is on the inner. And we'll discuss in a minute, these are key regulatory points. Once you've swapped your fatty acid onto carnitine, you get a fatty acyl carnitine, which can then go through the translocase located in this inner mitochondrial matrix, give you that fatty acyl carnitine in the mitochondrial matrix. Now we want to get the carnitine back. The carnitine is basically going to, the fatty acid is going to be swapped back onto our CoA to give us a fatty acyl-CoA. And then this carnitine is going to be sent back through so it can pick up another fatty acid. From one molecule of palmitoyl-CoA, you could actually generate a lot of energy. So from the breakdown, we are getting the 7 FADH2 and the 7 NADH, and then the 8 acetyl-CoA. And then we're taking that through the TCA. We're getting 8 GTP, 8 FADH2, 24 NADH, and then all of that could go through the electron transport chain. So lots of energy. And this was just coming from a single fatty acid. And remember that often that's going to be coming from something like a triacylglyceride. And so you're going to get the glycerol as well as all those fatty acid tails. And so fatty acids really do provide a large source of energy because they have all that oxidative potential. So let's talk a little bit about regulation. Fatty acid synthesis and breakdown are going to be inversely regulated. So if we're making lipids, well, when do we want to make lipids? Well, we want to make lipids if we have plenty of energy and more than we can handle right now. Let's store some. If we're wanting to store lipids, we're not going to be wanting to break down lipids at the same time. Similarly to how we wouldn't want to be doing glycolysis and gluconeogenesis at the same time and breaking down the glucose as we're making it. We don't want to be breaking down our fats as we're making it. And so we want to have the products of this fat making be inhibiting the enzymes that break down fats. the easiest way to inhibit the fats from being broken down, the most efficient way, is to keep them from getting into the mitochondrial matrix, which is where that beta oxidation was taking place. So remember how milano-CoA, that 3-carbon intermediate we got from carboxylating acetyl-CoA, that was going to be produced during our lipid synthesis. We can have milano-CoA negatively inhibit our CAT1 or CPT1, this thing that was going to take that fatty acyl-CoA and basically hook it on to carnitine to get it into the mitochondria. Rather than taking these groups into the mitochondria, we want to stop that, and so milano-CoA is going to inhibit CPT1. Another key point of regulation is this ACC, or this acetyl-CoA carboxylase. That was how we actually produced the milano-CoA. When would we want to stop this from happening? Well, we would want to stop this from happening if we were building up a lot of fatty acids. If we're building up a lot of fatty acids, maybe we need to slow things down. Use that acetyl-CoA in other ways, maybe. And so we have these fatty acids, so like our palmitoyl-CoA can actually inhibit this ACC. When would you want to stimulate the ACC? Well, maybe you want to stimulate it if you have a lot of citrate. So if you have more citrate than, say, your tricarboxylic acid cycle can handle, or basically you have citrate and you don't need as much energy, what you want to do is basically you want to send the message down the line, hey, get ready for us. So the citrate is going to positively regulate the ACC. So when we have a lot of citrate, that's going to stimulate the generation of these fatty acids. Remember too that citrate was a negative regulator of glycolysis by inhibiting phosphofructokinase 1, PFK1, which was that key point of dedication into the pathway of glycolysis. It makes sense that we're kind of reciprocally regulating fatty acid synthesis and glycolysis because, well, if we have a lot of energy and we're wanting to store it as fats, we're not going to be wanting to break down sugar for energy. So if we have a lot of citrate, let's stimulate our lipid synthesis and let's inhibit glycolysis. We also saw how long-chain fatty acids, such as our palmitoyl, such as that palmitate we made, that can actually go and inhibit pyruvate kinase. So pyruvate kinase, remember, this was taking our phosphoenol pyruvate to pyruvate in the process of glycolysis. And so basically, we would want to inhibit it. So we're inhibiting glycolysis in this way, promoting instead the change of pyruvate into that oxaloacetate, and that we can then take through the citric acid cycle in order to get our acetyl-CoA that then we could use to make these fats. So we see coordinated regulation between the different pathways. Another point of regulation is they're like the lipases that are actually going to cut down the tags into fatty acids. And so this can be regulated by things like hormones, various signaling pathways. And so this is all going to be tightly regulated. The fuel from fatty acids, remember, we can break things down to acetyl-CoA, but we couldn't transport the acetyl-CoA that way through the blood. And so instead, what we're going to want to do is we're going to want to convert that acetyl-CoA into things that we can transport through the blood. Especially, this is important for your brain. Your brain's really picky and it won't break down fat for energy. And so if your brain runs out of its preferred food source, it's glucose, well, then your liver needs to send it energy, and it does this in the form of these ketone bodies. These ketone bodies come from acetyl-CoA, but they're not acetyl-CoA. Instead, we have acetoacetate and beta-hydroxybutyrate, as well as acetone, which is just a non-enzymatic byproduct, which isn't the one that you want. Instead, the main form that we have as our carrier is going to be this beta-hydroxybutyrate. It's not technically a ketone body. It's got a carboxylic acid group rather than a ketone, but it can get converted into acetoacetate, which is a ketone, and that can be used to generate this energy. Basically, you can get two acetyl-CoAs hooked together through a couple of different enzymes to give you acetoacetate, which can then get converted into beta-hydroxybutyrate, which is just the reduced form of it. So acetoacetate, it can spontaneously decarboxylate to give you acetone, which isn't what we want. And that's because it has this beta-keto acid. Beta-hydroxybutyrate is a reduced form of this, so we reduce this to an alcohol. We don't have that issue. This is going to be more stable, and we can transport it through the bloodstream. One medical connection notes, this you might hear about this HMG-CoA synthase, so that's responsible for joining together these two acetyl groups, acetyl-CoA groups, to give you this acetoacetyl-CoA. There's some intermediates before you get to acetoacetate, but this HMG-CoA synthase is the one that does this initial step. And it's also, this is going to be important for making steroids and making your cholesterol. And so HMG-CoA synthase is actually a target of statin medications for controlling blood cholesterol. But it's also an important step here for producing these ketone bodies. These ketone bodies can then be transported from the liver to tissues like the brain in order for the brain to then use them, assuming that it has enough tricarboxylic acid component still in order to do so. Another medical note is acetone, this spontaneous byproduct that we don't want. Basically, it can form if you have a lot of these ketone bodies building up because there's not enough of the components of this TCA cycle for that acetyl-CoA to enter and be broken down to be oxidized fully. These ketone bodies can build up in the bloodstream. It's just a condition called ketosis. Sometimes you'll see like ketoacidosis. What happens is because if we look at the acetoacetate and the beta-hydroxybutyrate, those have carboxylic acids. They're going to acidify the blood, leading to this condition where you have acidified blood caused by these ketones, which is ketoacidosis. You see that sometimes with diabetes that's uncontrolled. And you'll also see that their breath is going to smell like nail polish remover or floral scent type weird fruity scent. That's from the acetone, that bad product that can build up if we can't be handling the things great. Because remember, with diabetes, what's happening is the cells aren't taking up enough glucose. The blood sugar can get high, but also you don't have enough energy inside of the cells. So the cells are starving, even though there's going to be plenty of glucose around. It just can't get in. And the glucose was being broken down in order to give us our tricarboxylic acid components that we could then use to break down, to oxidize things fully, to give us energy. If we aren't able to go through the TCA, well, then we're going to basically have this acetyl-CoA build up. If that acetyl-CoA builds up, well, now it's going to go do alternative things. We could make fatty acids with it, but that wouldn't be very helpful for us here because, well, what we need is energy. We're not trying to store fatty acids. And so instead, we get this problem, we get this build up of these ketone bodies. And so that's often a diagnostic sign of uncontrolled diabetes, is having that like acetone breath. Fats aren't the only way that you can get these ketone bodies. They can also be produced by some of the amino acids. So this brings us to our topic of amino acid metabolism. In order to break down an amino acid, you're going to have to take off the amino group and then deal with the parts. Sometimes when you take off an amino group, you're going to get a component of the citric acid cycle. And we call these amino acids glucogenic. Because basically, if you have a component of the carboxylic acid cycle, that can then get used to make glucose. In order to do this, however, you need to have get broken down into something that's going to be greater than 2 carbons, because you're going to in the cycle you're losing 2 carbons each time. So breaking something down to acetyl-CoA isn't going to be enough to make something glucogenic. That's going to, because you're adding 2 carbons and you already had to have these carbons in the cycle in order to get things going. So on net, you're not going to be able to make glucose from it. For those amino acids that get broken down into acetyl-CoA or acetoacetyl-CoA, those can then be our ketone bodies. They can enter the tricarboxylic acid cycle if there's components present, but they can't be used on net to generate glucose. So we'd say that those are ketogenic, and some of them are going to be both. All of it depends on what you're left with when you remove that amino group. Amino groups are removed through a process called transamination. Often what's going to happen is the amino group from one amino acid is going to be added on to alpha ketoglutarate. This will give you an alpha keto acid and glutamate. So it's an alpha keto acid. We've got a ketone in the alpha position to the carboxylic acid group. So before we're talking about a beta acid, keto acid, this is an alpha keto acid. And which alpha keto acid you're left with is going to depend on what amino acid you started with. And often these are going to be components of the tricarboxylic acid cycle, or components that can be converted easily into one of them. This happens with the help of these enzymes called transaminases, which typically use a PLP cofactor, which I'm not going to go into the details of here, but I have more if you're interested. Basically, you can take any amino acid, and almost any of them has a transaminase that can transfer the amino group from it onto alpha ketoglutarate to give you glutamate. Your cells do this strategically so that then they just have to deal with glutamate rather than having to have more enzymes that are going to deal with each of those individual amino acids separately. Instead, we get this central source of glutamate that then we can regulate that and use that glutamate to transaminate different things or to dispose of the waste safely as urea. So basically that nitrogen, we don't want the nitrogen to just get chopped off and float around because that could be free ammonia is dangerous to your body. Whereas if we take it and we can take this glutamate and we controlledly release that nitrogen group in the liver, we can use this urea cycle, which is then going to take that waste and dispose of it safely. But we can also take that glutamate, that amino group, and transfer it onto different things, which will allow us to do things like interconvert between different molecules. Remember that we could take basically any amino acid and make glutamate. Well, now what we can do is we can take that glutamate and we can transfer the nitrogen onto something else. And so we can take that nitrogen and transfer it onto pyruvate to give us alanine, or of course we could go the other direction as well. In these two cases, we have alanine transaminase or ALT and aspartate transaminase or AST. You might have heard of these called like liver enzymes. Basically, these enzymes are in your liver and they play important roles. And when your liver is damaged, these can leak out into your blood, and then doctors can measure that as a sign of liver damage, as well as like heart damage. What are these important for? Well, it turns out that your body, when they want to transport these nitrogen groups through the bloodstream, they don't actually do it in the form of glutamate. Instead, the main ones that transport through the blood are going to be glutamine and alanine. Imagine that your muscles are working really hard. They run out of their glycogen. They're going to then go and break down protein. When they break down protein, they have those nitrogen groups, but they don't have the urea cycle to dispose of them safely. Instead, they're going to send those nitrogen groups through the bloodstream, where they can then be put onto alpha ketoglutarate to give you glutamate, which can then go through the urea cycle to give you that urea. Alanine transaminase is going to be important for that step, and so you'll have it in your muscles as well as your liver. The muscles get a really sweet deal from this because now the liver, it's actually going to take that pyruvate that it generates, and because the liver also can do gluconeogenesis, it's going to convert that back into glucose that the muscles get back. So the muscles are transferring 3 carbons and this nitrogen group, and they're getting back 6 carbons and don't have that nitrogen group to worry about. So pretty sweet deal. And that was our ALT. AST is also going to be really important. And this is important. That's going to be our taking oxaloacetate and making aspartate. Aspartate can enter the urea, is a component of the urea cycle. So you're getting one of the amino groups from like glutamine or glutamate, and then you're getting another one from this aspartate, which can then enter the urea cycle. Doing this transfer is going to happen by this aspartate aminotransferase. And so this AST is really important for getting rid of that waste safely. Arginine is also going to be an important component. It's an intermediary in there where basically you can have If you imagine cutting this part off, but with an oxygen, well, now you're going to get urea. Amino acids coming into play all over the place. Like just as we saw before, how if we wanted to make aspartate, or we want to make glutamate, or if we wanted to make alanine, we could do that by using tricarboxylic acid components. Well, similarly, we could make many of the other amino acids. We can make some of these amino acids from other ones, different ways that we can synthesize these, and then we could use them for different things. One of the things that methionine is really great for example, is for transferring methyl groups. So methionine, remember this is its structure, it's got this thioether group. So basically you have this sulfur and it's got this methyl group on the side. We talked a bit about sulfur. We said it was really great. It's got this big cloud. It makes these weak bonds. We can swap things on and off. And so we can swap this methyl group off. Now, if this was normally super, like if this methyl group could just like come and go, that wouldn't be very good. So instead we want to do it controllably. And the way that we can do this is by basically activating it. We can activate it by adding it on to this to adenosine. So adenosine triphosphate, or ATP. Normally we see these phosphate groups get transferred. Maybe we see an AMP or an ADP transferred. In this case, we're actually just seeing this adenosine group get transferred. We're getting the hydrolysis of this ATP, so we're still getting energy that way. We're using energy that way. But here what we're doing is we're attaching this adenosine. When we do this, we're going to be activating this sulfur to be even more vulnerable, this sulfur carbon bond to be even more vulnerable, because the sulfur is now positively charged. It's going to be draining the electrons even further from this carbon, making this carbon very electrophilic and subject to attack by a nucleophile. What's going to happen now is you could have all sorts of different nucleophiles attack, and then you're going to go from having an unmethylated thing to having a methylated thing. And now you're left with basically this molecule, this adenosine homocysteine. So when we called this methionine plus adenosine, this was S-adenosylmethionine or Adomet or SAM. You might have seen all those different terms. It's a common like cofactor that's going to be used in various reactions where you're wanting to methylate something with the methyl transferase. When you take the methyl group off of it, you get this SAH, this S-adenosyl homocysteine. But that can actually go and it can be used to make cysteine. And you can get the adenosine back and all that good stuff. You see that you have this kind of like N-5-methyl THF and this THF. This is actually coming from folate. You're transferring a methyl group from folate onto the SAH or onto this homocysteine. Now, that folate itself is the methyl group on that folate is probably coming from serine. You have this basically like serine to glycine. You lose this methyl group, which can get transferred to folate. Various versions of folate. Folate can be used for all sorts of one-carbon transfers, and that's all thanks to serine. What about the glycine that you make? Well, the glycine can be used to do things like make purines, make glutathione, a reducing agent, even make our hemoglobin or the heme part of it. So all sorts of different things that you can do with various amino acid parts. Other than just like break things down fuller for energy. Speaking of breaking things down and not breaking things down, well, we can actually add on to amino acids to make them more complicated, but like signaling molecules and things. So some of our hormones are actually going to be derivatives of amino acids, as well as some of our hormones are going to be longer peptides that you can get by like cutting up a bigger protein. An example of that would be insulin. But some of the hormones are actually just kind of modified versions of amino acids. A couple of common ones from tyrosine. We can actually make catecholamines, so dopamine, noradrenaline, adrenaline. And look here, we have our SAM playing a role here with this methylation step. So you see all this like cooperation between these two different things, these different amino acids. You can also use tyrosine to make your thyroid hormones. Other hormones made by tryptophan. So we get serotonin, melatonin, various hormones made by various amino acids. And this is just a small sense of things. So amino acids, fatty acids, carbohydrates, all of them produce all of these various kind of like interchangeable parts. The processes of making and breaking them can happen in different components. We get our fatty acid synthesis happening in the cytosol. We've got the breakdown happening in the matrix. We've got glycolysis happening in the cytosol. We've got tricarboxylic acid cycle happening in the matrix. We have all this compartmentalization, all this regulation, all this really, really cool stuff. A lot of it involves kind of amino acids as serving key roles as either donating parts, being broken down for parts, being built for parts, or at least being interconverted between parts. So I think that kind of viewing things from the light of amino acids provides a way for you to see how really everything is interconnected. So it's not so much that it's all about the proteins, but instead it's kind of just showing you like, yeah, all these little parts can be made from different things, can make different things. And so that's part of why metabolism breaks things down into these small little pieces, because then you can work with those pieces and mix and match them in various ways and have alternative sources for building things rather than having to rely on getting big parts of things.